Today we will take a look at the salient features of 24-hour chick embryo. So we will just have the important features in a nutshell. The first thing is the shortening of primitive streak, which extended up to the anterior region during gastrulation. It was during gastrulation that we saw the primitive streak. So once gastrulation is completed and once the embryo reaches 24 hours of age, we can see the primitive streak retracts and get confined to this area, to the posterior region. So that is one of the features, that is the shortening of primitive streak and the movement of the primitive streak towards the caudal region of the embryo, because gastrulation was the event that we discussed just before, where we could see that the primitive streak started from this area and it extended up to the anterior region, and mesoderm started involuting inwards. The notochord also started developing, and we saw various features of the primitive streak. And thereafter, towards the end of gastrulation, what we saw is that the primitive streak starts retracting, and once it reaches the 24-hour stage, we can see that the primitive streak is confined towards the caudal region of the embryo. So it is an indicator that gastrulation is almost completed, and it is indicative that the morphogenetic movements are also completed. So that is the first feature, that is the shortening of the primitive streak. Now we will go to the second one, that is we have already seen the chick embryo. When we started with gastrulation, we saw that the central area was called as the area opaca, the area pellucida, and the peripheral area was called as the area opaca. So this is the area pellucida, and the outer region is called as the area opaca. So for that we will go to a diagram which we saw during the gastrulation studies. That is, when gastrulation started, we saw that this was the disc-like embryo, and we took a section where we could see the central region below which the subgerminal cavity develops. It was called as the area pellucida, which appears lighter, and the peripheral region of the disc is called as the area opaca. So that was one of the changes that we noted during gastrulation. So once it reaches the 24-hour stage, these two areas, that is the central area pellucida and the outer area opaca, here you can see, so this is the central area pellucida and this is the outer area opaca. So they are now distinctly visible. So once it reaches 24 hours, you can see distinctly visible area pellucida and area opaca. Now the third one. Third one is the differentiation of mesoderm. Do you remember? That is, how did the mesoderm form? The mesoderm started by involution of cells through this primitive streak inwards. There was a groove inside, and through the groove the cells started involuting inwards, and that formed the mesoderm. Okay. So now we will see what is happening to the mesoderm. So the mesoderm, which has involuted inwards, now differentiates into three regions. This is the first region. This is the first region, this is the second region, and this is the third region. So the first region is called as the dorsal mesoderm or the somoid. The second region is called as the intermediate mesoderm, and the third region is called as the lateral plate mesoderm. So somoid or dorsal mesoderm, intermediate mesoderm, and the lateral plate mesoderm. You can also see that the lateral plate mesoderm splits into a somatic layer and a splanchnic layer. So this is what happens to the mesoderm. We will just go to the previous stages where the mesoderm was forming. Here you can see this was a section through the primitive streak, and it was through the primitive streak, the primitive groove, that the mesoderm cells involuted inwards. The cells which are marked in black are the mesoderm cells which involuted inwards. This was ectoderm, this is endoderm, and you can see the mesoderm cells involuting inwards. Now afterwards, what you can see here, this mesoderm, this is the ectoderm, this is endoderm, and this is the mesoderm which is involuted inwards through the primitive pit. primitive groove, and this is the dorsal part, which is called dorsal mesoderm, second part called as intermediate mesoderm, and third part called as the lateral mesoderm, which is differentiated into the somatic mesoderm, which is lying close to the ectoderm, and the splanchnic mesoderm, which is lying close to the endoderm. Okay, so this is how the mesoderm differentiates. Now we will come to the somites. Somites are actually metamerically arranged blocks of cell masses. What is metamerism? Segmentation. So they are seen as segments. They are segmentally arranged, and you can see them as block-like cell masses. So this is a section. We will go to the direct diagram of the 24-hour chick embryo where we can see the somites. So look here. These are the somites. You can see them as block-like cell masses on either side of the embryo. So these are the metamerically arranged somites. Okay, so these somites are formed from the dorsal mesoderm. So you can see them as segments, as pairs of block-like cell masses which are segmentally arranged. Okay, so when you take a section, you can see this. Take a section, you can see the somite like this, which is followed by the intermediate mesoderm and the lateral plate mesoderm. Now we have to count the number of somites. In a 24-hour chick embryo, so when you take a 24-hour chick embryo, there will be 4 pairs of somites, in which this is the first somite. Okay, so the first pair of somite, it is formed at the 21st hour of development. So it is during the 21st hour of development that the first pair of somite is formed, and the remaining somites will be formed posterior to it, because if the first somite is formed here, the others will be formed behind it. So the remaining somites are formed posterior to it. So during which hour is the first somite formed? It is during the 21st hour. Now, after the first pair is formed, in every 1 hour, a new pair gets differentiated. Okay, so the first somite is formed in hour 21. So the second pair will be formed in hour 22. The third pair will be formed in hour 23, and the fourth pair will be formed in hour 24. And we are studying about the features of the 24-hour chick embryo. So we can see 4 pairs of somites in the 24-hour chick embryo. So we can just guess the number of somites in 33-hour chick embryo. The first one is formed in 21st hour. So the 13th one will be formed in the 33rd hour. At 48-hour embryo, you can see 28 pairs. Okay, so first pair at 21, second pair at 22, third pair at 23, fourth pair at 24. So the 13th pair will be developing in the 33rd hour. Okay, so the counting of somites will help you determine the age of the embryo. So that is the third feature, the development of somites. So we saw the shortening of primitive streak, we saw the distinct area opaca and the area pellucida, we saw the 4 pairs of somites. Now we will go to the next process, that is beginning of neurulation. What is meant by neurulation? Neurulation is the formation of neural tube. When you take a section, you can see this is the neural tube. It is from this neural tube that the nervous system will develop. You are already familiarized with neural tube formation in frog embryo. So likewise, we will just come here. So this is an earlier embryonic stage where gastrulation just started in chick embryo. You can see the primitive streak forming. In a later stage, you can see the primitive streak is advancing and it is ending at the Henson's node. It is ending at the Henson's node. I told you at the end of the primitive groove or primitive streak will be the Henson's node, and arising from the Henson's node will be the notochord. All these things were discussed during gastrulation. So just in front of the Henson's node, you can see the notochordal process developing. Okay. Now we will go to the neural plate formation. At 18 hours, the neural plate starts forming. That means this part of the ectoderm is destined to become the nervous system. So this part of the ectoderm is destined to become the nervous system. It is called as the neural plate. So this is formed at around 18 hours of development. Okay. So this is formed at around 18 hours of development, and you can see an advanced stage where you have a distinct neural plate, a thickened area of the ectoderm which is called as the neuroectoderm develops into a plate-like region which is called as the neural plate. And by that time you can see another phenomenon. The primitive streak is retracting. Earlier the primitive streak was advancing, but now the primitive streak is retracting and the neural plate is distinctly visible. Now what will happen to the neural plate? The neural plate will sink down and two faults will be formed. When it sinks down, it will start developing neural faults. Okay, it will start developing neural faults, and the neural faults, they finally come together, converge and form a neural groove, and finally it will develop into a neural tube. The same events which took in frog neurulation, that is the neural plate formation, and at the edges of the neural plate will get folded to form neural faults. The neural faults start growing towards each other and finally they will fuse to form the neural tube. Okay. So this is how neurulation takes place. So the neural, you can see the neural plate formation started at the 18th hour, but neural faults are formed during 24 hours. And they extend from the anterior end of the primitive streak forwards. So the faults are formed from here and they advance forwards. Okay. So that is a fourth feature, that is beginning of neurulation. Now we will go to the next feature, that is the differentiation of notochord. So what is notochord? If this is neural tube, there will be a rod-like structure just below the neural tube. It is called as the notochord. So notochord is the rod-like structure which is formed just below the nervous system of the neural tube. So how is it formed? Do you remember in one of the classes in gastrulation we saw that this is a lateral view, but this is a cross-sectional view. So this diagram is familiar to you, that is you can see this is the primitive streak, this is a lateral view. Okay. So this is the primitive streak which extended up here, and this is the primitive pit, and this was the Henson's node, and it is from this Henson's node that the notochord started developing. Did I tell you? The Henson's node, from this Henson's node the notochord starts extending forwards. So we can say notochord arises as a median outgrowth from the undifferentiated cell mass of the Henson's node. So from this Henson's node it will arise as a median outgrowth. Okay. So this can be compared to this area of origin of the notochord or the Henson's node is equivalent to the dorsal lip of the blastopore of frog gastrula. In frog gastrula also we saw that it is from the dorsal lip that the notochordal cells started involuting inwards. So here also it is from the Henson's node that the notochord starts developing. So you can consider it to be equivalent, not similar in structure but equivalent to the dorsal lip of the blastopore of frog embryo. So if you are asked a question, that is the area in chick embryo which is similar in function to the dorsal lip of blastopore with regard to notochord development, you can answer as the Henson's node. Okay, because it is from the Henson's node that the notochord starts developing. Okay, we will just go to another diagram. Here this is the primitive streak, this is the Henson's node. These are different views. Okay, so this was the primitive streak, this is the Henson's node, that is the end of the primitive streak, and you can see the notochord started developing from the primitive streak to the anterior region. That is what we just saw. This is a view from the dorsal side of the embryo, and this is a view from the lateral side. Okay, that is why there is a difference. So from the Henson's node you can see the notochord extending forward. Okay, and what is this extending mass called as? The extending mass is like a slender process. It is called as the head process. Okay. So at the end of, this will start soon after gastrulation, and at the end of 24 hours, the notochord will assume a definite rod shape. So that we can see in this diagram. You can see the notochord has assumed a distinct rod shape at 24 hours. You can see here. So, the fifth feature of 24-hour chick embryo is the differentiation of notochord from the Henson's node as a slender process which is called as the head process. So, this is the notochordal head process which finally develops into the notochord. Okay. Now, the next one. Next one is the formation of head fold. So, we will go to what is a head fold. Here you can see this region which is like a horseshoe. You can see this like a half moon, this region like a half moon, this region. It is called as the head fold. So, simultaneous with the formation of the neural fold, the anterior part of the embryo, here we will just again come to the neural fold. This is the neural fold, okay. This is the neural fold, and this is the groove inside the neural fold. This is neural fold, okay. And we will come again. So, simultaneous with the formation of this neural fold, the anterior part of the embryonic area will get thickened. Here you can see this structure which is like a crescent or a horseshoe. This structure, it gets thickened and it elevates above the level of the blastoderm. It elevates above the level of blastoderm with a well-defined anterior boundary. You can see here it has got a well-defined anterior boundary which will appear like a crescentic fold, which will appear like a horseshoe. This will become the anterior border of the future head of the embryo. So, this will become the anterior border of the future head. And there is a space between the developing head and the blastoderm. I told you this head fold develops a little elevated above the remaining blastoderm. That was just said earlier. That is, the head fold gets thickened and elevated above the level of blastoderm. So, there will be a space between the head fold and the remaining blastoderm, and that space is called as the subcephalic pocket. That will be seen inside below this. So, the region between or the space between the head fold and the blastoderm and the remaining blastoderm is called as the subcephalic pocket. Okay. Now, we will go to the next feature, that is the seventh feature, establishment of foregut. What is meant by foregut? It is the anterior part of the alimentary canal. Okay. Both the ectoderm and endoderm are involved in the formation of the head. So, when the head process is forming, the endoderm in the interior region is also getting folded. So, I told you when the head is forming, it is not only the ectoderm which is taking part. I told you the ectoderm is forming the head fold, but the endoderm is also undergoing a pocket within the ectoderm. Okay, this pocket you can see, this is the outer region which is called as the head fold. In the interior, you can see a pocket formed by the endoderm. This forms the margin of the foregut, or this endodermal pocket, it will become the foregut or the anterior part of the alimentary canal of the embryo, and it is the first part of the digestive tract to acquire a definite cellular floor. So, that is the first part which will acquire a definite cellular floor. Okay. So, this is what is called as head fold formation and foregut formation. Head fold is formed from the ectoderm like a thickened and crescentic area above the remaining blastoderm, and interior to the head fold you can see the endoderm is also forming a pocket, and it is this endodermal pocket that will later form the first part of the alimentary canal, that is the foregut. Okay. So, the endodermal pocket becomes the foregut of the embryo, and it is the first part of the digestive tract to acquire a definite cellular floor. Okay. Now, the posterior, this is the foregut, and this is the anterior region of the foregut, and this is the posterior region of the foregut. So, this is anterior region, this is the posterior region, this region is the posterior region. It is inner. Because of the presence of this neural fold, you cannot see it, but interiorly you will have this pocket which will form the foregut, and the posterior region of this foregut, okay, it will open to the midgut. by a wide aperture, and that is called anterior intestinal portal. They are all inner structures which cannot be represented in this diagram. So you have to imagine, that is, a pocket is formed here, that will form the foregut, which is followed by the midgut, and the aperture which is separating the foregut and the midgut is called as the anterior intestinal portal. Though it is marked here, it is an inner structure. It is a structure which is seen towards the interior. So the head fold formation and the foregut formation takes place simultaneously. Even though they were discussed as separate points, the events took place simultaneously. Then the next one, the next feature, that is the eighth prominent feature which you can see in a 24-hour chick embryo, that is the formation of blood islands. Look here, these are blood islands. You can see blood islands here. So what are blood islands? In which area are they formed? The central area is the area pellucida and the outer area was the area opaca. You can see here, this is the area pellucida and this is the area opaca. Okay, this is area pellucida, this is area opaca. It is in area opaca. You can see aggregates or small clusters of mesoderm cells. So these are small clusters of mesoderm cells which are, they are all clusters of mesoderm cells which are accumulated in the area opaca, and they are called as blood islands. So what do they mark? They mark the initial step in the formation of blood vessels and corpuscles. So the formation of blood islands is indicative of the initiation of formation of blood vessels and blood corpuscles. Okay. Then the next is the formation of coelom. So what is coelom? Coelom is the body cavity. If gut is the inner cavity, there will be a cavity surrounding it, that is called as the coelom, and usually it is formed from the mesoderm. So we will just see what is coelom. So for that we will go to this diagram, that is here. Look, we earlier talked about the mesoderm. This was the somite or the dorsal mesoderm, this was the intermediate mesoderm, and this was the lateral plate mesoderm. Okay. And I told you the lateral plate mesoderm is differentiated into an outer somatic layer and an inner splanchnic layer. Okay. You can see a space between. It is this space which will become the coelom. So by extension, this there will be a widening of this space and this will develop into the coelom. Okay. So we will have a look. This is ectoderm. This is endoderm. And this is mesoderm. But just remember, this is only a part of the embryo. The embryo will be like this. Embryo will be like this. And there will be the gut here. The gut will be here. The ectoderm will be here. And in between, so you have a central gut which is lined by this endoderm. Remember, this is only a portion of the embryo. If it is a full embryo, it will be like this. The endoderm will be forming a gut here. The ectoderm will be forming a covering here. And the mesoderm will also be like this, it will be full, and this space it will widen and form the coelom. So the coelom appears in between the somatic and splanchnic layer of the mesoderm. It is this area. Actually the coelom consists of a pair of bilaterally symmetrical chambers, one on this side and the other on this side, and these two chambers are bilaterally symmetrical, but later they become continuous. The coelom on this side and the coelom on this side, they will become fused and they get continuous. Okay, this is the coelom on the right side or right side of the embryo, and this is the coelom on the left side of the embryo, and they fuse together, they become confluent. Okay, now when the space widens, the somatic layer will become close to the ectoderm and the splanchnic layer will move close to the endoderm. Okay, so when this coelom widens, the somatic layer will go close to ectoderm, the splanchnic layer will go close to the endoderm. Now, this ectoderm plus somatic layer is called as the somatoplure, and the endoderm with the splanchnic layer is called as the splanchnoplure, and the space in between will become the coelom. Okay, so this somatic layer plus ectoderm becomes the somatoplure, and the splanchnic layer plus endoderm becomes the splanchnoplure when they move towards the ectoderm and endoderm respectively, and this will lead to the widening of the space, and this space forms the coelom. Okay. So, we just now discussed the coelom. Now, the coelom, which is, so the entire embryo will have a coelom, but the coelom at this region, the coelom at the anterior intestinal portal region. So, imagine this, there is a tube inside which is the gut, and there is an outer tube which is the coelom. You can just imagine a pen with a refill. The refill is the gut, and the cavity around it, the space around it is the coelom. So, coelom will be extending whole throughout. The coelom has already started forming, but this coelom at this region, the coelom at the anterior intestinal portal region, they undergo widening. The coelom at this region undergo widening and they develop into the pericardial cavity. So, the coelom at this region undergoes widening and develop into the pericardial cavity. What is meant by pericardial cavity? That is the cavity in which the heart is located. Okay. So, and now what happens? At this region, the splanchnic mesoderm will undergo thickening and give rise to the heart primordia from which the heart is formed. Okay. The splanchnic mesoderm. So, this at the intestinal portal region, the coelom dilates to form the pericardial cavity, and the splanchnic layer of the mesoderm will undergo thickening and give rise to the primordia of the heart. Okay. So, the heart is formed from the splanchnic layer of the mesoderm. Okay. So, that is how the pericardial region is formed. And the last feature in chick embryo is the differentiation of area vasculosa. So, we will go to that. I already told you, this is the area pellucida and this is the area opaca. Okay. So, area pellucida inner, area opaca outer. So, we will go to another diagram which we saw earlier during gastrulation. Do you remember? This is the area pellucida and this is the area opaca. This is a subgerminal cavity below which is the yolk. So, the part of the outer region of the disc-like embryo which is closely opposed to the yolk is called as the area opaca. So, area opaca consists of yolk just beneath, but it is beneath the area pellucida that you can see the subgerminal cavity. So, we will now just leave the area pellucida, we will just take the area opaca. What will happen to the area opaca? The area opaca differentiates into two regions. So, I will come to take you to this diagram. So, the central region is the area pellucida which is marked as A, you leave it. This is area pellucida, okay, the same area pellucida, okay, you just leave it, we are leaving it. We are going to this area opaca, okay, area opaca. What is happening to the area opaca? It has differentiated into two. This inner layer or this proximal layer is called as area vasculosa, and this outer layer or distal layer is called as area vitellina. So, where will you find more yolk? This region will be having more yolk beneath, because when you go outer, you will see more yolk. This area opaca itself is differentiated into an area vitellina and area vasculosa. Okay. So, area vitellina, inner area vasculosa and inner area pellucida. So, this is area vitellina, this is area vasculosa and this is area pellucida. Area pellucida was already there. It is area opaca that differentiates into area vitellina and area vasculosa. This vitellina is having yolk beneath. This area is having yolk beneath, just beneath it there is yolk. This area vasculosa is a region where there will be extension of mesoderm, because the suppose this is the primitive streak. You just imagine this is the primitive streak. It is through the primitive streak that the mesoderm cells start involuting inward, and the mesoderm is involuting inward and extending to this area. The mesoderm is involuting inwards and it is extending to this area, and because of the extension of this mesoderm to this area will be darker. It will appear darker because of the entry of cells to that area, and it will have a mottled appearance like this. You can see a dotted or mottled appearance. This area is the area vasculosa. So, this dark and mottled appearance of this area is because of the extension of mesoderm through the primitive groove. Okay. And this area is called as the area vasculosa. So, it is from this area vasculosa that the blood islands develop from mesodermal tissues. Earlier we discussed about the blood islands. Okay. And the outermost region or the distal region of the area opaca is called as the area vitellina, beneath which is yolk concentrated. Okay. So, these are the various features of a 24-hour chick embryo. So, we will just discuss again in a nutshell, that is the shortening of the primitive streak, distinctly visible area opaca and area pellucida, the differentiation of mesoderm into somite, intermediate mesoderm and lateral plate mesoderm, splitting of lateral plate mesoderm into somatic layer and splanchnic layer, then beginning of neurulation, we saw here the beginning of neurulation, the neural plate formation at 18-hour and neural fold formation at 24-hour. Then the differentiation of the notochord, you can see the extension of notochord from the Henson's node and it develops into a rod-like structure. Then we saw the formation of the head fold, the crescentic area and the foregut which is formed from the endoderm as a pocket. Then we saw the formation of blood islands here, the blood islands, these are the blood islands from mesodermal tissues. Then the formation of coelom here by the widening of the space between the somatic layer and the splanchnic layer, and the coelom at the anterior intestinal portal will dilate to form the pericardial cavity. And last one is the differentiation of this area opaca into an outer or distal area vitellina and an inner or proximal area vasculosa. Area vasculosa is rich in mesodermal tissue, that is why it is having a dark and mottled appearance. Area vitellina is lighter because of the presence of yolk beneath. So, this finishes the salient features of the 24-hour chick embryo. Thank you.